Hello biology class. We are continuing with chapter 18 on evolution of populations and looking at molecular evolution right now. So this is the evolution of genes in a very simplified way. So far in this chapter, we've talked about the evolution of the genotype of the organism and the gene pool at large. So we've been tracking how allele frequencies change and whatnot. Now, natural selection tells us that it is nature that selects for the traits, but modern genetics taught us that genetics shapes the individuals. Darwin in his time knew nothing really about genetics, certainly nothing about DNA. Nobody in the world did at that time. So books like The Selfish Gene, which I have that right here, I've read this, by Richard Dawkins. He wrote that way back in 1976. That sort of that book is really why he's famous. It revolutionized the way we think about evolution. So our focus really should be on the genes themselves rather than the entire organism, because the genes are what shape the organism. And changes, like as we see species evolve or subspecies evolve, the changes are extremely incremental. They're small changes from generation to generation. And yeah, over a long period of time, a whole brand new species pops up, or the differences between species become very great. But it's really just small changes at the gene level that occur. You don't get 500 mutations all at once, and then that produces the new species. No, it's way more gradual than that. All right, so humans, we have around 25,000 genes. E. coli, which is a bacteria, it only has around 3,000 genes. Drosophila, which is the fruit fly, they have around 15,500. So where do additional genes come from? And then as we get new genes, how do they get modified for new purposes? Because not only do we have more genes than flies do, we do things that flies can't do. Like our body physiology is totally different. We have a skeleton, for example. They don't. Well, one way this occurs is through gene duplication. So if you remember from crossing over, homologous chromosomes, in your body chromosomes that you got from each parent that have the same genes, well, they exchange part of their genetic material. So gene X on one chromosome swaps and for the same gene on the other homologous chromosome. But sometimes a chromosome receives part of the other chromosome, but it doesn't give back. So it has an extra copy of that gene. So here we see two homologous pairs, one that this person inherited from their mom, one from their dad. So we have genes A, B, C, D, E, F, G on them. They're the same genes, but because they were inherited from two different people, they're slightly different, so I made the colors slightly different. In crossing over, the chromosome will flip-flop some of those genes. So we can see that genes EFG on one side of the chromosome got swapped for the other side. We can see that they're no longer exact copies. They're the same genes, but not exact copies of them. Well, sometimes when that happens, like here, we can see that the one chromosome took genes EFG, but it only gave back genes F and G. So now it has two copies of E. And theoretically, in the future, one of those E genes could be modified for a very slightly different function. And then, who knows what that would cause, but that is a source for new genes, acquiring new genes, and acquiring new characteristics and traits by modifying those genes. So as I was saying, over time, those duplicated genes, they can evolve to have a different function. And we know for a fact that this happens because in humans, for example, we actually have 6 copies of the same gene. It's a gene for making one of your hemoglobin proteins. So you only need one, but we have 6 for whatever reason. And there are tons and tons of other examples of this, but we don't really need to go over every one of those. And then there's genetic rearrangement. Now, we actually covered this a long time when we studied DNA and chromosomes, but the the genes on a chromosome can be rearranged. Instead of A, B, C, D, E, F, G, it can like go backwards, for example. There can be a recombination with another chromosome, a non-homologous one. Or genes can, two different chromosomes have their genes expressed, and then those genes combine with each other later on during transcription and translation. There's a huge number of ways in which genes can be rearranged. It's really complex. It would take forever to go over all of the details of this, but it's well studied by thousands of biologists over the years. And the numbers of ways that genes can be expressed, this has helped make eukaryotes way more complicated than prokaryotes are, because they only have one chromosome, they don't do sexual reproduction, etc. So here are just some illustrations, examples to show you how a chromosome can be altered. We see the duplication, that's the one we already looked at, but you can have a deletion. That doesn't really add material, it subtracts it. But an inversion can make the genes express in a different way and then maybe force them to be repurposed over time. And then the translocation is sort of like crossing over both non-homologous chromosomes. So you get genes from two different places all of a sudden interacting together, and that can produce totally new changes. Something we've also discussed before are the Hox genes. So these are organizers. They determine which parts of an embryo will develop into legs, arms, wings, etc. Hopefully you remember what we've learned about these in the past. Groups of Hox genes, which I think are called homeotic genes, they determine the size and shape of those structures. So you have the same Hox genes that a fly does, but we also have many additional ones because we share common ancestry with flies, but it's from an extremely long time ago. We are more complex than they are, so we've accumulated more changes. And small changes, really small changes in Hox genes can end up having very dramatic effects to the body plan of the organism. So it's not the case that one organism will give birth to another organism that has like 50 or like 500 brand new mutations, and all of a sudden, holy crap, it can do like things we've never seen before. No, the mutations are usually much smaller in number, but occasionally they can have a very large effect. So the thing with Hox genes is they line up on the chromosome from head to tail, which is kind of weird. So if a gene is behind another gene, like on the chromosome, that'll mean that that gene codes for a part of the body that is below that part. So like here we see the fruit fly, its genes, they're color coded based on what part of the body that gene ends up affecting. And then on the bottom, Hox A, B, C, D, that's humans. We have extra ones, but they still line up exactly the same way they do with flies. They create different structures in us, we have more of them, but we share a whole bunch of them. So an example that your book gives is if we take arthropods. Arthropods are all the organisms with an exoskeleton and segmented bodies. So this includes crustaceans, insects, and what are called myriapods, which a centipede or a millipede would be. Insects only have 6 legs. Myriapods will have a whole bunch. Crustaceans, they have 5 pairs of legs. So if you look at the lobster, it's got the big claws, that's 1 pair, and then 4 additional pairs from there. Now all insects only have 6 pairs, or 3 pairs of legs, 6 legs. It turns out we found the gene where a mutation on this happened. It's called the UBX gene, and it's what has basically caused insects, their whole group diverged, and yep, we have 3 pairs of legs, and we're just fine with that. Whereas the crustaceans kept their 5 pairs of legs. And then there's a little section called timing is everything. So as an example of what they mean by this, all mammals except for sloths and manatees have 7 neck vertebrae. So now a giraffe, extremely long neck, but still only 7 neck bones, just like you or me. So our neck is only this long, a giraffe neck is like, whatever, 8 feet long or something. They grow that long neck by having that part of the body active during embryonic development for more time. And this even shapes the subtle differences between you and me. So if you have like a really large bridge on your nose, or if you have like really long or really short fingers. I kind of have like wide hands, but short fingers. Big hands, big feet, larger or smaller ears. All this stuff is really just down to timing. How long was the Hox gene active for? And that can mean two people that are the same height have like radically different sized hands. Their body proportions will be totally different. Maybe one has really long legs versus the other. You get the picture. I have this really awesome book called The Sports Gene, and it's all about biology, genetics, and sports. So over time, we've turned athletics into a science, and we've figured out exactly the best way to throw the javelin or the discus, the exact best way to tackle somebody in football. And what we have found is people of different shapes and sizes fit the mold for their particular sport really well. You take the greatest Olympian of all time, Michael Phelps, with his like 20 gold medals or whatever he has. Well, he has actually kind of a weird body. He has a really, really long torso and short legs, and he's got an incredibly long wingspan. Now, long arms, that's pretty obvious it would be advantageous in swimming, but a long torso and short legs is not exactly as obvious. Michael Phelps, who I think is like 6'4", he has the torso of an average person who is like 6'8", way taller than him. But his legs, so like when he goes and shops for pants, he buys the same, his inseam is the length of somebody who's only 5'9". So his legs are way disproportionate to his torso, and both of those are disproportionate to his overall height. People who throw the discus, you have to have your arms straight out to launch that discus. So we've seen by measuring people that, yeah, the best discus throwers in the world, they have like really long arms. Coaches in like really serious programs, especially like track and field, because there are so many different events you can do. You can throw things, or you can jump, or you can run. Really serious coaches will look at their athletes and they'll kind of like measure out their proportions and be like, you know what? You would actually, I know you've never done this before, but you would be a great high jumper. You have the body for it. And then these people start doing it, and then all of a sudden they just start to break records and whatnot. So that's a really cool book. If you like this kind of stuff, I recommend taking a look at it. But again, those body shapes differences that we all have, two people that are the same height can still have like totally different body proportions. It's all down to these Hox genes, and timing is everything. It's impossible, mutations are random. It's impossible to know when a mutation will occur, but it is possible to know roughly how many mutations will occur over a certain period of time. So it's really hard for me to think of an example of this, and I have to use kind of a morbid one, but we take like deaths, right? Now we don't know when anybody really is going to die, but we do know like every month or whatever, X number of people will die, right? We can statistically basically figure it out. We don't know exactly who is going to and exactly when, but over a long enough period of time, we can pretty much figure out how many of those events will occur. And it works with mutations too. So for example, humans and chimpanzees, we are around 2% different genetically. We share 98% of our genes. Well, a question that biologists want to know is, how long did it take to accumulate that 2% difference that we have? And what about an animal that we are 50% different from? How long did it take to accumulate that much difference? So theoretically, if we could figure out how long it took to make that 2% difference between humans and chimpanzees, let's say it's like, it ends up being like 5 million years or whatever. Okay, well, then we can take that and multiply it by the 50% difference, and that's how long ago we branched off from this other animal, right? That's actually not really how it works. I'll be more specific here in a second, but that's just kind of an example. So, molecular clocks, they use mutation rates in DNA to estimate how long ago two species diverged from each other. Molecular clocks are based primarily on small mutations that don't necessarily affect your phenotype. Every one of these small mutations is like a click on the molecular clock, so it's like the second hand as it goes around. And by sequencing genomes of different organisms, we can see how many clicks or ticks or whatever you want to call them have occurred between those two organisms. And then we can try to guess how long it took to get that many clicks, and then we can go backwards and say this is when they branched off from each other. So if we look at this chart, this shows just like change over time in a gene sequence. So we have the original gene sequence all the way on the left, and then it branches because two mutations happen. So for the purposes of this, we're going to say that that is when two different species evolved. So we have the G on the one that used to be a T, it looks like, and then we have the A on the other that used to be a C by the looks of it. So those two strains of DNA are 90% similar to each other. And then the one on the bottom, that goes on its own evolutionary pathway. It accumulates another mutation, another A, and then that one accumulates two new ones. We have the G and the T, right? And then on the top we have another G, they're in blue, and then that species branches off into new species that develop their own mutations, a purple C and a green A, for example. So in all of these, if we go all the way to the right, we can see that there are three distinct ticks or mutations in there. So we would want to see how long did it take to accumulate those, and then how long ago was this original ancestor of all of these organisms. Using molecular clocks is extremely complicated, and we're not really doing it justice here today. It's really complicated because mutations happen at different rates for different organisms in different places. We can't compare the entire genome of one organism to the other because mutations in one part of your genome accumulate faster than other parts. So what do I mean by this? Well, here's an example. Let's say we take two random people and we want to figure out how old they are, but we can't ask them like, what year were you born? That'd be the easy way. So we can look at these people and be like, okay, well, we have to figure out how do people change over time? Okay, well, could we do like how tall they are because people get taller over time? Well, no, because one of them could be 7 feet tall, like Shaquille O'Neal. He was 7 feet tall when he was a high schooler. And the other person, maybe they're like only 6 feet tall. But that doesn't tell you how old they are, right? So that doesn't work. Could we look at how long their hair is? Well, your hair gets longer as you grow, or as you get older, but people cut their hair, so it can't really tell you anything. But if we stick with the hair, there is something maybe we could do. Maybe we could count the number of gray hairs, because that, although different people get gray hair at different parts of their lives, that's a fairly good estimate for, okay, how old is this person? Count the total number of gray hairs they have. Now, is that scientific? No, not really, but it's just an analogy, okay? So much like your hair grows faster than you do, and your nails grow slower than your hair does, and all that stuff, all those things are wildly different, but the rate at which our hair turns gray, that's a lot more consistent. So we only want to compare this one thing. What we do with molecular clocks is we compare mutations in a single gene that we know the two organisms share. And that's what we track the differences in. So we don't count the overall like 2% genetic difference between us and chimpanzees. We pick a gene that both us and chimpanzees have, and we see how much it has changed over time. Now we have to calibrate our molecular clocks too, and one way we do that is with the fossil record. So like we use the molecular clock method and it says, okay, well it says that they should have branched off with each other 10 million years ago. But we look at the fossil record and we see, well, we have tons and tons of fossils and we see that they became distinct only 8 million years ago. So we scale back the molecular clock a little bit because nothing's perfect. But science is a process of solving problems. No other method has a way to do this. We can calibrate, we can fix our scientific methods to get them to tell us what the truth about things is. So that's it. Short section, but a lot of fairly complex details. We'll have a couple other things on Google Classroom, or I'll put up a couple more videos for you to check out this week to help you investigate these things. And in the meantime, have a good rest of your week and take care.